Hello class, welcome to our first virtual lesson of Unit 5. Here's today's agenda. Number 1 is the do now. Before you get into these notes, make sure you finish that do now on Canvas. Now, let's go and grab a piece of paper and pencil. Since I can't provide you Cornell notes, you will have to copy this on a separate piece of paper that you can reflect back on. Now, just some key strategies about taking notes. Only copy the things down that will help you understand. There's no point in needing to copy everything down. Some of it is just so that you can better understand it when you read it. So in this case, only write down the things that I have underlined for you. But if you feel like you need to write more, pause the video and write yourself an explanation. After these notes, you'll be completing a web quest that is again found on Canvas that'll help you understand this topic better. Then you will do a summary, basically answering 3 essential questions, and after that you will do a mastery quiz. Now, here are your essential questions for the day. First thing, okay, so your first question is, why do cells reproduce? What are the 3 phases of interphase? And what is the correct order? What is it keep doing that? What is the correct order of cell division stages? So make sure you copy these 3 down, and you will be answering them in the discussions part on Canvas later on. So make sure you copy these down so you can type this out later. I will be looking for this to grade your work. And I apologize for my computer. I don't know why it keeps switching slides by itself. So the first question I pose to you all is, who in the class has reproduced? Now I want you to think about the idea of what reproduction is. Reproduction is the creation of life. Now the first thing that probably popped in your head is, who has created a baby? But I want you to think about what the smallest living thing on earth is. It's not a baby, and it's actually cells. And your cells, your body cells, make more and more copies of itself every day. And if cells are a living thing and they're making more living things, isn't that reproduction? Now, so technically everyone who is watching this video has reproduced. Maybe not in the sense that you're familiar with, but in the new way that we're going to learn what reproduction is. Now the next question is, why do cells reproduce? So there are 3 reasons. 1, to make more cells. 2, to repair and replace them. And 3, to maintain a workable volume to surface area ratio. Now, in layman's terms, all that means is to stay small. Now we're going to review these 3 things and go into more depth. But first thing, let's look at this. To make more cells. Again, this goes back to our Stirner process with growth and reproduction. In order for organisms to get bigger, they need to get more cells. Remember, larger organisms don't have bigger cells. They just have more cells than smaller organisms. For example, Dwayne "The Rock" Johnson is a lot bigger than me or anybody in this classroom. Now that's not because his muscle cells are bigger. It's just because he has more muscle cells than you and I. Or maybe your favorite basketball player is taller. That's not because he has bigger bone cells. It's because he has more bone cells than all of us. All right. Second. That's not what I had in second. Give me a minute. To repair and replace cells. Now, why do we want to repair and replace cells? First, let's think about it. If you get cut or injured, you have a big gash. And maybe a couple days later, you will see something about that changing. You'll see that the cut's smaller. Maybe it's become pink. This is because cells, your skin cells, are making more copies of itself. So that way they can close and basically fix that part of the body. And honestly, even if you're not injured, your cells will still replace itself because it's getting old. It's not functioning anymore. So in fact, your skin cells replace itself daily. And actually by the time that you're 32, you no longer have the same cells that you had when you started as a baby. So it's basically like you are a whole new person. Now, finally, the more complicated reason to maintain a workable volume to surface area ratio. Now, all this means is that your cells need to stay small. The reason why is because it makes it easier to move things in and out of the cell, right? To move toxic, unnecessary things out, and to move things that we need in and around the body. For example, remember when we talked about the very tall high school basketball player who was about 7'6"? We said he had a hard time um, moving blood through his body, but this is because he was too tall, right? Too big. If he was a little bit smaller, he would have an easier time doing that. Here's another example. Think about our classroom, right? Let's say you wanted to pass a note to your classmate without me seeing. Now, when it would be easier to pass that note? When the area is smaller and you're right beside each other, or when the area is large and you have a big distance between each other? And your answer should be it's when the desks are right beside each other and the area is smaller. Okay? So in summary, those are the three things of why do cells divide. 1, to make more cells. 2, to repair and replace cells. And 3, so that cells stay small. Okay, that should be your first answer to essential question 1, and that's what I should be seeing in our discussions page. Second, do all cells divide and reproduce the same way? The answer to that is no. Basically, this is because cells divide in 2 ways. There's a prokaryotic division and eukaryotic division. Now, the difference is one happens quicker than the other one. Pro means no nucleus and no mitochondria, while eukaryote, U means they do have a nucleus and do have mitochondria. That's the only thing different. Now, what they have the same is that cell division produces always 2 daughter cells, and it's always going to have exact same DNA. But that is called asexual reproduction. The 2 types of reproduction you're going to learn today is one, sexual, which is what you're already familiar with. It's when a male and a female have sex and make a baby. Now, asexual reproduction, the word asexual means without sex. Without sex means that organism can make a baby by itself. But this means it's going to have the exact same DNA. Now, real quick, let's do a quick review of prokaryote, eukaryote. Again, pro means no nucleus. All it needs to do is copy the DNA and then make another cell, and then boom, it's done. While eukaryotes is a lot more complicated process because they need to replicate the nucleus, DNA, and other membrane-bound organelles. So this is going to take a lot longer cycle. Now, let's get into the new things. Cell cycle and mitosis. This is going to be the bulk of your test, so make sure you understand this very well. So the first thing we're going to talk about is this G1S and G2. So make sure you write this down. All this is parts of the interphase. Interphase, as you can tell, takes a majority of this circle because the cell spends 90% of its life in the interphase. And these 3 stages are G1, S, and G2. G1 is when the cell grows and it increases in size. So you can think about it as G1 standing for growth 1. S stands for synthesis, where DNA is copied. Again, you need to make copies of that DNA in order for that cell to be the same. And G2 is when the cell prepares for mitosis. So we have growth, replication, and then prepares for division. All right, and the second part you need to know about the cycle is mitosis. Mitosis, to keep it simple, P stands for prophase, M stands for metaphase, A stands for anaphase, T is telophase, then finally you have the final step, cytokinesis. Now I like to just call this PMAT, PMAT, for all phases. Alright. And just a heads up, you'll probably be seeing these terms, so I don't want you to become lost when you do see them. Chromosomes versus chromatin, all of these, both chromosomes is just DNA that's tightly packed, while chromatin is loose DNA that looks like noodles. Think about it this way. When you have a ramen packet, or some of you might know as oodles of noodles, think about what it looks like before you boil it. When it's hard and in that package, think about that as chromosomes. But after you boil it and it's loose, that's chromatin. So that's the difference between those two things, but it's still made of the same material, DNA. Now, let's take a quick look at what prophase, metaphase, anaphase, and telophase mean, because you do need to understand what they look like and what's in them. So I would go ahead and make a quick chart, because you will be drawing and writing this in a piece of paper. The first step again is interphase. We talked about it has three phases, G1, S, and G2. Now here's what you don't know. The picture is just going to basically be two circles, and inside the smaller circle you have the noodles, because here DNA looks like chromatin. While we have next is prophase. This is the first step in mitosis. The chromosomes start becoming visible. So the noodles, the chromatin, they start coming together, condensing, and now they look like X's. That's what we call chromosomes. Additionally, you start seeing the inner circle disappearing, and you have these spindle fibers forming. Now those spindle fibers come from the centrioles that are only in animal cells, and they look like thin pieces of string. So that's prophase. Second step, metaphase. Now notice I underlined the word M. The reason why is this is when the chromosomes line up in the middle. This is an easy way to remember it. When the chromosomes are in the middle, that's metaphase. Think about it. MM, metaphase, middle. Next step is anaphase. Again, I underlined the A. If you notice, the chromosomes in the middle now are being pulled apart. Anaphase starts with the letter A, apart starts with the letter A, so this is an easy way to remember it. Anaphase, the chromosomes are being pulled apart by the centrioles. So that covers I, P, M, A. Now let's cover T, telophase. Step 4 in mitosis. The phase of mitosis is when the nucleus reappears. So you start seeing that inner circle happen again, and the two cells that are identical are almost split up. Not all the way, but they're almost there. That's all. That's really the visual that you need to see. And finally, cytokinesis. Oh, excuse me. The final phase in cell division, where the cytoplasm separates and the cells go to opposite poles. So now all you have to do is cut that little part off, and now you have two identical cells. Now here's the final thing you need to understand. Cytokinesis in plants versus animal cells. Animal cells are going to have a cleavage furrow right here, while plant cells are going to form a cell plate. Because remember, plant cells don't have they don't have centrioles, so they can't actually do the same things that animal cells do. So that's the main difference. The furrow right there, where you see that pinch, while cell plates just have, in plant cells, they have a cell plate, which is a line. All right, that is it for our notes. Make sure to go ahead and go to Canvas and complete this cell cycle and mitosis worksheet, and I will be checking for that later on. Now I know you have other classes to complete, but complete as many modules as you can. Ideally, hopefully you can get one lesson done a day, maybe two. This is probably the most difficult lesson, so really do your best to take time and understand this, because understanding the rest of this lesson is to do that, you need to understand this first one. Okay? Again, I will have office hours between 9 a.m. and 11 a.m. and 12 p.m. to 2 p.m. daily. So please come visit. Let me know. And I will see y'all in the next video.